Hello, hello. Before we get started, once we start, could everyone please refrain from walking in front of that camera? Thank you very much.
All right, you guys, you all ready to get started? All right, so the 8:30 section, when is your final? Wednesday in 2 days. 7:30, when is your final? Yeah, on Monday. So we're going to start out looking at chapter 5. One of the things that people don't realize is really important in chapter 5 is the different types of epithelium you have for different organs. Each plays a specific role in what the organ does and its function. So I made this chart, and I really liked this chart when I studied for the class. So we're going to go through it and see if I can help you guys a little bit. So our first organ is the urinary bladder. So I condone you guys kind of yell out answers if you know them. So urinary bladder, what type of epithelium is it? Transitional. What's another name for transitional epithelium if you didn't see that on your multiple choice? Yeah, urothelium. All right, so what's something special about the transitional epithelium or urothelium? What makes it different? And you guys are free to discuss. Feel free to turn to the person next to you and see if they have any ideas. So I'll give you guys a couple seconds to do that. Just think for a minute about what makes urothelium a little different. All right, so does anyone have any ideas? It can stretch? Yeah, so, and here's what I brought these papers for. So it has a lot to do with the apical layer of cells. So for example, urothelium typically is cuboidal in shape. But when your bladder is distended, which means it's full of urine, the apical layer of the cells are going to smush down like an umbrella or like a muffin. So that's what makes urothelium a little different. And it's because your urinary bladder is typically filled with fluid. So here we're going to say that it's compressible. All right. Good. So now what about the male urethra? So we have different parts to the male urethra. Let's start with the most internal, closest to the urinary bladder. What type of epithelium will you guys see there? Urothelium? Yeah, so we're going to start out with urothelium because we're starting closer to the urinary bladder. All right, so after urothelium, what's the next epithelial transition? So I heard stratified columnar. Does that sound right to everybody? All right, so a little important detail. It's pseudo-stratified. So we have pseudo-stratified columnar cells. All right, what comes after that? Yeah, I heard some people say squamous. So then we transition to squamous or squamous, however you feel the need to say it. So urothelium, we're going to see that in what's called the bulbal portion of the urethra. They just basically combine the prostatic and a little bit of the preprostatic. Sorry, I said that backwards. The preprostatic and some portions of the prostatic are urothelium. There, a majority of the spongy urethral area is pseudostratified columnar. After that, at the ampulla in glans penis is what will have simple squamous columnar. All right. So what about our prostate gland? What's the epithelium type for the prostate gland? Say that again? So in your, I skipped one. All right. So in your book, it says prostate epithelium in quotes. All right, what's a little more detail we can add to that? Good, so we start out with urothelium. Urothelium is pretty common. All right, anything else you guys have to add? All right. I made this box a little too small, so I'm going to write here. So for the prostate, we also said basal cells. What type of epithelium do basal cells have? Cuboidal. A little specificity, low cuboidal. All right, anything else? Good, we have mucus cells. All right, so what's something special we see here in the prostate? Acid secretions in the prostate. So in the prostate, we see mildly acidic secretions. The way I like to remember that is prostate starts with P. P, sort of like proton. If you guys remember from chemistry, acidic solutions have a high concentration of dissociated protons, which are just hydrogen ions. So P, proton for prostate. Prostate has acidic secretions. All right, excellent. So actually I skipped one. So let's go back to the seminal vesicles. What type of epithelium will we have there? Pseudo-stratified columnar. All right. What's something important we have, special characteristic of the seminal vesicles? Basic secretions. We have alkaline secretions. What else? Yeah, that's a really important thing. Here, we also have fructose. What's the purpose of fructose in these secretions? Yeah, they provide energy for the sperm. Prior to the secretions of the seminal vesicles entering with your ejaculatory fluid, sperm cells are non-motile. But we'll get to that later. All right, so moving on to ovaries. What are the epithelium type for ovaries? Simple cuboidal? All right, sounds good. Simple cuboidal. All right, moving on. So what about the uterus? All right, so the apical layer is called the endometrium. And I heard a couple people say that this is simple, high, columnar. We'll talk a little bit more about the epithelium when we talk about menstruation. All right, so the fallopian tubes. Yeah? Okay. Okay, I'm just going to move that over here. I ran out of space. So, the vaginal portion. Good. So, stratified, non-keratinizing epithelium. This was similar to epithelium in what other organ? Yeah, awesome. The esophagus. All right, so the fallopian tubes. Fallopian tubes is pretty similar to the uterus. Yeah, we still have simple high columnar. The only difference is that in the fallopian tubes is where we have the deposit of the ovum. So that's the mature egg cell. So we will need cilia. Cilia will cause, yeah? All right, so vaginal, we said stratified, oh, I forgot to write squamous, thank you. Stratified squamous. All right, thank you for catching that. All right, and we already talked about it up here, so let's repeat it. Vaginal epithelium. I want to hear everybody say it. Yeah, there we go. Stratified squamous non-keratinizing epithelium. All right, so let's talk a little bit about the epididymis. Can any of you guys name the epithelium types in the epididymis? It starts with a little bit of a funky word. So when we start out naming it, we call it stereociliated. Pseudo-stratified. Columnar epithelium. All right, so how do you guys feel about the epithelium in the organs? If we find anything else that's important for epithelium, we can always come back to this and add to it. Because this is only from chapter 5. In some of the other chapters, we did discuss epithelium. Some of it's more important than others, but we'll come add it back to this section if we find anything else important. All right. So continuing on with chapter 5. What my intention is for this exam review is to go through each chapter and hit some of the topics that I know most students have the most trouble with understanding. So I'm not typically going to touch blood supply. I'm not really going to touch lymphatics because that's something you can read and understand on your own. So I'm going to hit the weird topics that are kind of not too mentioned that are really important to understand. And also a lot of things that students forget are important. So starting out with chapter 5, we have the pelvis and the perineum. One of the structures we have is the levator ani. It's a series of muscles. It's composed of 3 different muscles. Okay, so the first one is being the puborectalis. Do you guys remember the other muscles for the levator ani? I heard ischiococcygeus. Yeah, there you go. Iliococcygeus. I like how some of you guys were like, oh no, not ischiococcygeus. All right, what's another one? Yeah, pubococcygeus. All right. What a lot of people get levator ani confused with is the pelvic diaphragm. You will see some similarities. Spelled that wrong. All right, there we go. Pelvic diaphragm is also mainly composed of 3 muscles. And you'll see why it's easy to get these confused. One of the first main muscles is also the puborectalis. All right. Similar to the levator ani, we'll also see the iliococcygeus. Now here, the main difference is instead of pubococcygeus, we'll see what? Ischiococcygeus. Good. These are two pretty simple structures that a lot of people forget to look at. So you could see a question with lists of 4 different muscles and say, which of the following is not part of the pelvic diaphragm, or which is not part of the levator ani muscle group? Are you guys good on this so far? Good. We're only like 20 minutes in. So let's keep going. All right. I'm going to re-hit some of the organs that maybe have some small details we might have forgotten. So discussing the urinary bladder. What is the main muscle surrounding the urinary bladder? Yeah, detrusor. Now, at approximately what volume of urine will you feel the urge to micturate? Yeah, so it's like, it's a weird range. Usually we approximate about 300 mils. That's usually a safe range to go in the textbook. It's anywhere from like, I think 250 to 350, if I remember correctly. So we usually approximate 300. All right, so now we can talk about one of the few organs we'll talk about innervations on. Urinary bladder. The main innervation for this one is pelvic splanchnic. The roots being S2, S4. Good, and as you look into it more, you can see how parasympathetic and sympathetic will actually balance each other out. While sympathetic will relax the voluntary sphincter, parasympathetic will control, sympathetic will control your involuntary sphincters. Good. That explains why when you're running, you typically don't really feel like you have to micturate or urinate until you stop. And then your parasympathetic can come back into play. But besides that, urinary bladder, not too much else going on. All right. One of the other important topics that a lot of students will get stuck on, and this is where we start to talk about homologous structures. Right underneath the bladder is what? Yeah, I heard someone say it's the prostate. So here we're going to talk about the prostatic urethra. All right. So in here, you're going to see some different structures. We're going to talk about what they are, what they come from, and what their purpose is. All right. So here we have the dilatation in the prostatic urethra. Do you guys know what these two little circles here are? Say it again. Entrances of the ureters? All right, so that's why I wanted to hit this. These two are actually the ejaculatory duct openings. Up here in purple, what's that called? It's considered a blind opening. Yeah, good, the prostatic utricle. It's blind because it's not a real opening. It doesn't open into anything. Prostatic utricle is actually a remnant of Mullerian duct, which is why it's blind. It serves no purpose in the male reproductive system. All right, and down here, this region is the seminal colliculus. And this is where you'll have approximately, I think it's 20 to 30 ductules. Those open and are relatively important in regards to prostatic secretion. It's important to understand where these things are located so you can understand where these secretions empty into. Not to mention here we have a homologous structure. Prostatic utricle is remnant of the Mullerian duct in the male. Mullerian can go by another name. Do you guys remember what it was? Paramesonephric. Do you guys all agree? All right. All right, good. So we discussed the male urethra a little bit right here. The main important topics for the male urethra are to know the epithelium types, know these homologous structures, what go into these openings. Yeah? This says paramesonephric. Good question. Yeah, if you guys can't read anything, feel free to ask me to say what it is. Or if you want me to zoom in or zoom out. All right, so we discussed male reproduction. I'll just get a new paper. All right, now we're going to talk about female reproduction really quick. One of the important topics Dr. Sam told you guys to know in regards to the female reproductive cycle is menstruation. All right, this is a topic that's very easily overlooked. So I'm going to make a chart for you guys. Menstruation is broken up into how many different cycles or how many different phases? Yeah, it's about 3. So the first cycle is from day 1 to day 4. This says name right here. All right. Do you guys know what this phase is called? Yeah, so it's the phase of, and this word's always hard to say, it's dissemination. Did I say that right? I'll just run into here. All right. Yeah, just ignore that one. All right, so some characteristics about this phase. What happens in this phase? Let's talk about hormone levels. What happens to the endometrium? So which hormone goes up? Estrogen increase. All right, what about the other hormone, progesterone? What happens to it? Good, decrease. All right. Now, in this stage is actually where we see the endometrium is shedding. So I have DESQUAMATION. No problem. All right. So menstruation is regarded as beginning when we have the menstrual bleeding, which is the shedding of the endometrial layer. So the next set of days is days 5 to 15. Do you guys know what that phase is called? Yeah, so one of the names is ovulation, but it's also called the phase of proliferation. All right, so in this phase, let's talk about which hormone predominates. Yeah, good, estrogen. So estrogen predominates. All right. So here we also have the functional layer of the uterus grows. Functional layer refers to like the stratum compactum, where you see a lot of those blood vessels. And make note that a picture of the layers of uterus is going to be on your exam. I'm not sure if it'll actually be the picture itself or if it'll be their content based, but it's something that Dr. Samsam said to know. All right. Another important thing is during this phase, body temperature of the uterus. Body temperature increases by 1 degree Celsius. All right, so the next section is days 15 to 28. What's the name of this phase? Yeah, secretory phase. All right, so here, this stage is predominated by which hormone? Progesterone. So in this stage, progesterone must increase. And this leads to an increase of mucus secretions. It also leads to an increase in blood vessels. So blood vessel formation. And then the cycle starts over. From here, when progesterone drops and estrogen begins to increase, you'll actually see ischemia of these blood vessels. That layer of epithelium will then die and shed. And that's when you see the bleeding and menstruation. All right, any questions about menstruation? Yeah? All right, good question. So FSH and LH. What does FSH stand for? Yeah, so follicle stimulating hormone. What does LH stand for? Luteinizing hormone. These have a lot to do with the actual release of the ovum or of the egg into the fallopian tubes. This is covered mostly in physiology, so you won't really be asked too much on this. I'd say read over it once or twice, but as far as the actual functions of these hormones, you guys don't need to know. Does that answer your question? Okay, no problem. Yeah, it's good to know what they stand for and maybe know which phases they're high in, but even that is pushing physiology a little too much. All right, any other hormones for menstruation? Or, sorry, any other questions for menstruation? All right, so we can move on. Sticking with female reproductive, I want to ask you guys a question. I don't know if you guys can read this, but Cole's fascia. What's another name for Cole's fascia? I'll write this a little neater. right, so Cole's fascia is the equivalent to the superficial perineal fascia. These double-sided arrows means you can use either name. Now, one thing I will say, though, is that from this fascia is when you see the formation of two other fascias. And these will sound kind of familiar to you guys. You guys remember Camper's fascia and Scarpa's fascia? Which one of those is fatty? Campers. Yeah, because campers eat a lot of s'mores, so they get fatty. What about Scarpa's fascia? Is it fatty tissue or membranous tissue? Yeah, it's membranous. It's tougher. I remember that because these first few letters, scar. When you have a scar, bless you, when you have a scar, you degrade the elastic tissues and the elastic fibers in your skin. So that skin no longer becomes stretchy. It won't retract anymore. It becomes very stiff, like a scar. Scarpa's fascia is membranous. All right. You guys good on this? All right. So starting with topics we've seen before. Alcock's canal. What are the contents? I heard pudendal nerve. Anything else? Yeah, the pudendal nerve, artery, and vein. You can say nav or you can say van. Whichever naming system you prefer. Alcock's canal. All right. We're going to talk about the scrotum. How many layers are there in the scrotum? Scrotum has technically 6. You don't have to count one of them, but we want to be thorough. All right, we're going to start from external and go internal. External layer of the scrotum will be in purple. What is that? Yeah, skin. All right. So what comes after skin? Good. Dartos fascia. Dartos fascia and muscle. What's special to know about dartos fascia? Yeah, so it's thermoregulatory. This can cause the external layers of the scrotum to wrinkle and retain heat, or to relax if it's too hot. So here we're going to do that in orange. All right, so what comes after dartos fascia? Good. External spermatic fascia. I'm going to put that in green here. All right. After external spermatic fascia, what will we see? Yeah, famous cremaster muscle. All right, what comes after cremaster muscle? All right, awesome. Internal spermatic fascia. You'll sometimes hear people call this the cremaster sandwich. We have internal and external spermatic fascia surrounding the cremaster muscle. So we'll keep that in green. All right, what's the last layer? Yeah, awesome. Tunica vaginalis. So as long as you guys remember this cremaster sandwich, the only thing you'll need to remember are skin, which I hope you guys all remember you have skin. Dartos fascia. Dartos fascia is important because of what? Yeah, awesome. Thermoregulation, and then tunica vaginalis. Tunica vaginalis we learned a lot about in the last block. All right. Sticking with the topic of male reproductive organs. One of the pictures you'll see is the cross section of the penis. All right. Now to save you guys a little bit of misery, I'm just going to get the picture out of the textbook and not redraw it. People usually call this picture monkey face or raccoon face. Call it whatever you see fit. All right. So looking at these structures right here, this picture won't be in color in your exam. So a good thing to remember is that the largest venous structure right here in the center are the superficial and the deep dorsal vein of the penis. So what you want to do is start medial, move out, go superficial. Here you have the deep dorsal vein of the penis. Move out, have the deep dorsal artery, and then the dorsal nerve. So you have VAN. So you have dorsal van. And here you have the external or the superficial vein. All right, awesome. What's this structure down here? Urethra, specifically what portion? Yeah, good. Spongy urethra. So this muscular structure is called the corpus spongiosum. Be able to identify this is the penile urethra. And here is the deep artery of the penis. So they would be the eyes of the monkey. If I ask you guys to identify the tunica albuginea, what would you guys point to? So I'll give you some hints. Would it be this muscular structure in here? Would it be this membranous fascia right here? Would it be this fascia surrounding the corpus cavernosum? Yeah, good. This is the tunica albuginea, specifically of the corpus cavernosum. We also have a tunica albuginea of the corpus spongiosum. So as long as you remember tunica albuginea and what structure it's around, you'll be okay. cross-section of the penis is one of the more forgiving pictures. Any questions about this picture? No? All right, awesome. Good, so we'll move on. All right. All right. So now we're going to discuss some processes. Erection. Is erection controlled by a parasympathetic or sympathetic? Yeah, parasympathetic. Good. What about emission? Good, emission is sympathetic. Now, what about ejaculation? Yeah, awesome. Ejaculation is both. So here I have sympathetic. The sympathetic portion of ejaculation will close your urethral sphincter. This will prevent any urine from leaking or preventing you from actually urinating at this point in time. The parasympathetic will actually induce the peristalsis of your vas deferens. This is where the non-modal factor of sperm comes in. Up until the addition of fructose, sperm is non-modal. So the only way to move it from the epididymis to the ejaculatory ducts or to the seminal vesicles, where you will actually introduce the fructose, is through peristalsis of the vas deferens. So just to repeat that. Sympathetic closes urethral sphincter. I should be more specific, sorry. Internal urethral sphincter. This will prevent urination. Parasympathetic induces peristalsis of vas deferens. Oh yeah, sorry. Yeah, but you won't need to know this much detail. This just helps you guys realize how they kind of coexist. All right, so can you guys tell me what other pictures you will see from chapter 5 on your exam? We already looked at the cross-section of the penis. I can tell you guys that one you might see, the external genitalia of the female. So let's zoom in a little bit. All right. I left the names visible because we're just going to briefly go over this. So this picture will also be in black and white. You will not be able to distinguish anything based on color. So this lumpy structure right here in blue is what? The blue is part of the greater vestibule. That's not as important as what's right below it. This small little dot down here. Yeah, good. Bartholin's gland. Bartholin's gland is homologous to what structure in males? Cowper's gland. What's another name for Cowper's gland? Bulbourethral. What's another name for Bartholin's gland? Yeah, greater vestibular gland. That's 4 test questions right there, just from that one content. All right. I don't see too much else coming from here. Nothing that you guys didn't cover in lab. Maybe no perineal body. Perineal body serves a lot of the insertions for these perineal muscles. All right, any other pictures? Yeah? Okay, let's go back. Okay, so you had a question about emission and ejaculation. What was the question? Oh, that's all right. Okay. Thank you for that. So emission, again, like I said, this is detail you guys won't need to know. It's for your information. Emission is when you'll see the peristalsis and the secretion of like the pre-ejaculatory fluid. That just serves as like a lubrication and to neutralize the acidity of the vaginal canal. Yeah. Mm-hmm. Yeah, thank you for the correction. All right, any other pictures in this exam, or sorry, in this chapter that you guys think are relevant? Yeah? No, ejaculation is both. It is still both sympathetic and parasympathetic. The peristalsis, however, is more relevant to emission. Which is sympathetic. All right. All right, you guys feel ready to move on to chapter 9? All right, excellent. All right, so this is one of the pictures you're going to see on your exam in chapter 9. Let's talk a little bit about the structures you see. All right, so this first fold that's more anterior, what is the name of that fold? So I heard someone say palatoglossal. Do you guys agree? Good. Palatoglossal, it's forward, it's more towards your glossus, which is your tongue. What's the one behind it? Yeah, good. Palatopharyngeal. Again, this will be in black and white. If I had a pin pointing to this lumpy structure in between the folds, what would you guys call that? Palatine tonsil. Good. What about the dangly structure? Uvula. The space between the lips and your gum, what is that called? Yeah, vestibule of the mouth. What about inside your mouth? Inside of your mouth is oral cavity proper. All right, so this is an exam question coming from here. You'll see this picture on your exam. All right, another very important picture. All right, so what are these collectively? Yeah, good, they're nasal concha. So let's try and identify these muscles and these lymphatic structures. So these muscles here, you have 1, 2, 3 of them. What are they? These structures right here on the outside. You have 3 of them. They help squeeze the pharynx, squeeze these area. They're constrictor muscles. So you have your superior, middle, and inferior constrictor muscles. All right, here we have our uvula. This little thread-like muscle, what is this called? Yeah, good. The syringopharyngeus muscle. This thicker muscle right next to it, it's going at this angle. What is that called? Good. Levator villi palatini. Now, right here behind it, what is that muscle right here? Good. Yeah, you have the tensor villi palatini behind levator villi palatini. What about this hole? Good. Opening to the eustachian tube. Here we saw the palatine tonsils earlier, and here we see the pharyngeal tonsils. Good. So this is another picture mostly for identification. He will focus a lot on these muscles and their innervations. So there was one exception in this group. What was the exception to the golden sentence in this group? Tensor villi palatini. What's the innervation? Cranial nerve 5. Does that sound good to everyone? Good. Cranial nerve 5. What's the innervation of everything else? Pharyngeal plexus? Yeah, what are the nerves that make up the pharyngeal plexus? Good. Cranial nerve 9, 10, and 11. Which nerves or nerve make up the motor portions of the pharyngeal plexus? Good. 10 and 11. Which makes 9 what? Yeah, 9 is mostly sensory. All right. Those are the two most important pictures for this exam. All right. So let's talk about the salivary glands. Which salivary gland is the biggest? Yeah, the parotid gland. So what's the name of your other two salivary glands? Yeah, good. We have submandibular. And we also have sublingual. Parotid gland opens or drains into what duct? Stenson's duct? You guys all agree with that? All right, so we have that it drains into Stenson's duct. Where's the opening for Stenson's duct? Good. Superior second molar. If you stick your tongue up there and feel a smooth surface, that's the opening of Stenson's duct. All right. Excellent. So let's talk about the innervation for the parotid gland. What's the innervation? Let's start from the nucleus. Which salivatory nucleus will have innervation to this gland? Say it again. Cranial nerve 5. Cranial nerve 9. So is it the superior or inferior salivatory nucleus? So let's start there. So we're starting with inferior salivatory nucleus, which is typically associated with what nerve? Cranial nerve 9. Now, one of the more simple ways I used to remember this. Think about the series of numbers 7, 8, 9. 9 is more inferior to 7. 7 is more superior, and the word 7 also starts with S. So the superior salivatory nucleus is most commonly associated with what cranial nerve? Yeah, cranial nerve 7. All right. All right, so let's talk about the submandibular and the sublingual glands. Starting with submandibular, what's the name of the drainage? The structure it drains into? I think I heard someone say it. It's Wharton's duct. All right, what about for sublingual? Yeah, Bartholin's duct. From what it says in your textbook, Bartholin's duct will actually also join Wharton's duct. One of its few drainages. Sublingual will also have direct drainages into your mouth. All right, so I have a question for you guys. Are salivatory secretions similar to plasma? True or false? So I heard some people say true. If you say true, in what situations? You guys can take a minute and talk to the people next to you and just try to get an idea of the situation. All right, so you guys have any idea regarding if salivatory secretions can be similar to plasma? All right, so we're saying yes, only when it flows at a high rate. And the reason behind this is, there we go. So the reason behind this is when you have your salivary glands producing saliva, you're going to have various ions in there like sodium. You're also going to have lingual enzymes like amylase, lipase. So what happens is along the way, you have absorbing structures that will actually decrease the concentration of these ions, of these solutes. But when saliva begins to flow at a very high rate, there is actually not enough absorbing capability to reabsorb these ions. So at a high rate, soluble ions cannot be reabsorbed, which results in similar composition to plasma. You can think about it like an assembly line. If the assembly line is moving too fast, you won't be able to catch all the things moving across of it. Bless you. Any questions on this topic? All right, good. Staying relatively in the same area. We're going to talk about Waldeyer's ring. So Waldeyer's ring of lymphoid tissue is referred to as sort of the protective ring, or like the barrier. It protects your oral cavity from a lot of invasive pathogens. So let's talk about some of the structures that will actually compose Waldeyer's ring. You'll have the pharyngeal tonsils. They can also be called adenoid tonsils. You can also have tubule tonsils. This is located in the nasopharynx. Another common one, more commonly known, the palatine tonsils. And lastly, you'll have lingual tonsils. Good. Some of these structures you will actually be able to see on the posterior view of your nasopharynx. It's that picture we were just looking at. However, you won't be really asked to identify them. All right. Are you guys good on this? The only questions you'll probably see come from this are what lymphoid tissues actually compose the ring. Besides that, nothing too in-depth. All right. This was the topic briefly discussed in lecture. But we're going to talk about drainages of sinuses. We discussed this today in lecture. So we've only had one day to talk about it. But this is a very, very common source of questions. So here, I'm going to draw you something. Do you guys know what these are? Yeah, they're your nasal concha. You have your superior, middle, and inferior nasal concha. So to help you guys visualize this, I'm going to color in the bottom. Underneath each concha, we have what? Oh, yeah. There we go. Underneath each concha, we have a meatus. So here we have the inferior nasal meatus, middle nasal meatus, and superior nasal meatus. So while we draw this, I'm going to be writing down here. The various sinuses we have to worry about the drainages are the ethmoidal air cells. We have 3. We have our anterior ethmoidal air cells. Our middle ethmoidal air cells, along with our posterior ethmoidal air cells. All right. Do you guys know the drainage for the anterior ethmoidal air cell? Say it again. Can't really hear you. All right. So for the anterior ethmoidal air cell, we're going to have the forgot to add something. This is important. All right. So the anterior ethmoidal air cell is the semilunar canal or semilunar hiatus. So what we're going to do here, so up here I just wrote anterior ethmoidal air cell. And we're going to draw it in green, draining to here. This curved structure atop of the middle nasal concha is the semilunar hiatus. The middle ethmoidal air cells drain into what? Yeah, they drain on or above the ethmoidal bulla. You can't actually see the ethmoidal bulla from this angle. Ethmoidal bulla is located underneath the middle nasal concha. So the middle ethmoidal air cells will drain into the ethmoidal bulla. Relatively speaking, the ethmoidal bulla is more anterior than the semilunar hiatus. It's important to know the location of the bulla. All right, so what about the posterior ethmoidal air cells? Awesome. Posterior ethmoidal air cells drain into the superior nasal meatus. All right. So continuing on with the structures, we have the maxillary sinus. We have the frontal sinus. We also have the nasolacrimal duct. All right. So maxillary sinus, what's the drainage? Yeah, awesome. Maxillary sinus will drain into the semilunar hiatus. Maxillary sinus is, in your maxilla. Because of the cut of this picture, I can't draw it in, so I'm going to write it in over here. So maxillary sinus. Good. And we'll have this go also to the semilunar hiatus. All right. Frontal sinus. Frontal sinus is right here. It's in the front of everything. Frontal sinus drains into what? Middle nasal meatus. Good, so it comes around like this and drains here into the middle nasal meatus. All right, and the nasolacrimal duct. Nasolacrimal duct is also not shown on the image because of where the cross section is. The cross section is mid-sagittal. Your lacrimal ducts, do you remember where they are? Yeah, they're your tear ducts. So they're on the medial region of your eye, of your orbit. So we'll just keep it in the text. So nasolacrimal duct drains into where? Awesome. Into the inferior nasal meatus. Good. And the way I'll draw that is because of its location, I'll actually write it in up here. So the nasolacrimal duct. Good, and we'll have this drain into the inferior meatus. What's this pocket over here? This pocket over here is the sphenoid, or the sphenoidal sinus. Where does it drain? Yeah, so it kind of just drains atop of the superior nasal concha. The important part, though, is to know that this area that it drains through is called the sphenoethmoidal recess. This is the important part. The sphenoethmoidal recess. Yeah? Can you repeat that again? So the middle nasal meatus and the superior nasal meatus? Okay. Semilunar hiatus is a structure on the middle nasal hiatus is the best way to think about it. That's why I ask you guys to know relative location. If you think of the middle nasal concha sort of as like a cliff jutting out, for example, like this, if this is the actual concha, you could think of your ethmoidal bulla as a structure underneath, sort of like a little canal. Semilunar hiatus is more or less like a divot or like a hole on top of the middle nasal the middle nasal concha. You would put down the one it says in the text. You won't be asked to choose relative structure. So it's not going to show you a picture and say point to the ethmoidal bulla. Does that answer your question? Okay. Especially because you can't visualize the ethmoidal bulla for the angles of the cross sections he gives you. Good question. Do you guys have any other questions on this section? We will have a couple minutes afterwards. If there's anything you missed on these pictures, you can feel free to come up and take pictures of them or ask me about them. All right. Now, just to make sure you guys have been awake. What's the epithelium type here? Say it again. Non-keratinizing squamous. Do you guys agree? Right now we're looking at part of the nasopharynx and the nasal cavity. This is technically part of your respiratory tract. Repeat that. All right, this is something we talked about previously, the epithelium for the airways. I'm not going to make you guys say it now. Just keep that in mind. That could come up. All right, moving on to topics. I don't want to run out of time. We're going to go over chapter 10 really quick. There's not too much in chapter 10. And I'll save the cerebellum for last. So in chapter 10, one of the important topics is nerve conduction velocity. Oh, sorry. All right, so nerve conduction velocity, and know what attributes contribute to nerve conduction velocity. So what aspects of a nerve will make it send signals faster? So, yeah, I heard someone say myelination. So myelination will increase conduction velocity. What's another feature? Yeah, I heard someone say the size. So the larger it is, it'll increase conduction velocity. All right. So let's talk about the different nerve categories we have. We have A, B, and C nerves. Nerves in category A can be subtyped. Do you guys know the subtypes? Good. So starting, we have A alpha. We also have A beta. We have I always mess up my gammas. A gamma and A delta. So starting from which end would be the fastest conduction velocity? So A alpha, we're going to say is the very fastest. So which end would be the slower end? Yeah, so yeah, I'm talking about the whole list. So in the whole list, the C-type neurons are the slowest. So A, are A cells myelinated or unmyelinated? They're myelinated. They're myelinated and they're large. You don't need to know the exact measurements. So B cells, what do the B-type neurons have going for them? Good, so they're myelinated, but they're smaller. They're small neurons. What about C-classified neurons? Good, so C-class neurons are unmyelinated. And even smaller. All right, excellent. One other topic you might see regarding this chapter is reflex arcs. How many steps do you have in a reflex arc? 3 steps, do you guys agree? 3 steps for the reflex arc? That's something else. Repolarization and depolarization actually refer to the influx and outflux of sodium and potassium ions into the neuron or the axon. How many steps do you have in the reflex arc? Good, 5 steps. So, the first step we'll have is sensory reception. Good, so we'll have the actual stimulus. What's the next step? If you guys ever get caught up on this, what I think is the easiest is start with the beginning and go to the end right away. So we know the last thing that happens is you have the effector function. Good. So what do we call the sensory signal sent to our spinal cord? Is it afferent or efferent? Good, it's afferent. So we have the afferent fibers. So what comes after that? Awesome. We have the interneurons. Those are actually located in your spinal cord. All right, so then what comes after that? They exit the spinal cord. Good. Efferent or efferent. So then we have the efferent neurons. As far as reflex arcs go, you may need to know the steps. One of the important things to know, however, is that reflex arcs completely bypass brain signals. That's why reflexes are meant to be more self-preservation. So when you touch something really hot, the signal doesn't go to your brain and wait for your brain to tell the muscle to retract. It'll be recepted by your fingertips, sent to the dorsal root of your spinal cord, through the interneurons, out the ventral root, and that will tell the muscle to retract. It saves time, and then that helps preserve your life. All right. There's not too much on chapter 10. One of the other things I will stress, however, is the cerebellum. So on the picture of the cerebellum, you will be asked to name the lobes. Oh, I don't have it with me. All right. So the cerebellum is actually not in the first 25 pages of chapter 10. I don't have mine with me, so I'm going to draw a very rough sketch of a cerebellum. All right. So here, don't mind my art, but this is the brain stem. So looking at this here, we have midbrain, pons, medulla oblongata. How many lobes do we have to our cerebellum? Good, 9. We have 9 lobes. So let's go through them. I like to list out the letters first. All right. I personally think doing the letters, you can separate them however you see fit and make your own mnemonics. I'll tell you my mnemonic at the end. It's kind of silly. But our first lobe is what? Yeah, awesome. We have the lingula. The next lobe. Awesome. The central lobule. All right, after that. Excellent. We have the culmen. D. Excellent. Declive. F. Folia. Is it folia or folium? Folium. Awesome. T. Tuber. All right, what about the P? Pyramid. Excellent. The U. Uvula, just like in your oropharynx. N. Nodule. All right. So, it being Christmas, I was perhaps thinking about those people who like to save money on Christmas presents. So I split up the letters like this. Pun is its own word. Pun is something funny, so I left pun the way it is. But what I used to tell myself was that someone would buy a low-cost CD for the punny joke. Make up whatever works for you. If you want to use that, that's fine. Do you guys want me to repeat it, or are you good? All right, we'll just move on. All right. That's the extent you'll see of the cerebellum as far as its structure goes. Pay attention to the pathologies. If we have time, we have about half an hour left. I can quiz you on those really quick. I'm going to go through chapter 11 really, really quick. All right, so chapter 11. All right. Chapter 11, we're going to start with the eye section. So we talked about the lacrimal duct earlier. The lacrimal duct is kind of confusing in regards to its innervation. What's going to happen is you'll actually have the superior salivatory nucleus provide some of the innervation for this gland. Instead of duct, right gland. All right, so for the lacrimal gland, the innervation is actually going to start from the superior salivatory nucleus, but that is not the important part. The important part is that parts of the nerves will join cranial nerve 5. A majority of its innervation is by cranial nerve 5. So if you read your textbook, it talks a lot about beginning at the superior salivatory nucleus with roots of cranial nerve 7, goes into some stuff into the palatoglossal nerve. It joins with branches of the ophthalmic branch of cranial nerve 5. That is a majority of the innervation, and that's what counts as the innervation, cranial nerve 5. That is very important. Another structure you're going to see that's very important in this section is the Edinger-Westphal nucleus. All right. So, Edinger-Westphal nucleus is the origin of parasympathetic preganglionic fibers. For example, coming from this, you'll have the ciliary ganglia, or the ganglion. This is an important thing to know. The ciliary ganglion, you can see in the transitional cut, or sorry, I should say the transverse cut of the orbit. You can see the ciliary nucleus. You cannot see the Edinger-Westphal. It's important to know based on content. All right. Moving on. A very special hybrid muscle we've seen is the levator palpebrae superioris. What's special regarding its innervation? Yeah, so we said it's hybrid. So hybrid, it's 70% and 30% something else. 70% is parasympathetic. No, I said that wrong. Sorry, I'm getting myself mixed up. I was right the first time. It's sympathetic. 30% parasympathetic. By cranial nerve 3. All right. Another picture. So we're coming up to another picture topic. In this exam, the picture you'll see is the layers of the retina. The layers of the retina are not that hard once you realize the orientation of the picture. So when you look at the picture, you're going to see something like this. It's going to be split up into, I think, 9 different regions. When you look at the picture, the top is going to have some structures that look like this. What are these structures? Thank you. Don't be afraid to just like yell it at me. All right. So these structures up at the top of the picture you guys have on your exam, what are they? Yeah, awesome. They're optic nerve fibers. So this picture, here you're going to have the choroid. This is the basal area, meaning it's at the base. This is going to be situated in the eye like this. This being your choroid, and this being the optic nerve fibers. So when you see the plexes that you see, you'll see the outer and the inner. The outer is literally on the most outer side of the eye. So you can start here from the center and go out. We have our optic nerve fibers. We'll have the inner synaptic plexus. Moving on from there, you'll have various ganglia. And you keep going down. Knowing the orientation helps a lot with the picture. The last important thing I would say to know regarding the eye is blood supply. Main blood supply comes from ophthalmic artery, but ophthalmic artery has two very important branches. So the first one is the central artery. After that, we'll have the ciliary artery. This is what gets a lot of people. This top one says central, yes. Good. So like I said, I wouldn't touch blood supply too much except for the really important ones. This one's important and a lot of people forget it. Make sure to keep studying the continuation of blood supply on your own. So we're going to move on to the ear. All right. The picture you're going to see on your exam for the ear is the tympanic membrane. Know what it looks like pathologically. When we have it in a healthy person, split it up into 4 quadrants, where is the cone of light typically going to fall? Yeah, so about right here. It's where we'll typically see our cone of light. So just know where the cone of light lies when it's in a healthy tympanic membrane and when it's pathological. Do pay attention to the clinical points, especially in this part. They're always important, but this part has some pretty important ones. All right. So from here, we're going to move on to the internal ear structure. You have the ossicles, which are the bones. Now we're going to talk about the cochlea. The cochlea is also called the spiral organ. So it looks something like this, except I totally screwed it up. But cochlea. Cochlea has approximately 2.5 turns to it. When you unfurl the cochlea, you can see its structures a little better. So here, we have membranous labyrinth. And here we have bony labyrinth. One of the important things to know is where you'll typically have the reception of noises at what frequencies. So let's say you have a really high frequency, like 20,000 hertz. Will that be deeper or more towards the entrance of your cochlea? And there is a picture of this. Yeah, so a high frequency noise, I'm going to start drawing a diagram like this. So this is analogous to sound waves. This is analogous to sound waves. Very high frequencies are very high energy. So the way I usually think about it is, it's high energy, you want to stop it at the entrance. That way you don't injure the cells farther in your cochlea. So high energy, high frequency, they would be recepted more at the entrance of the cochlea. Low frequency, low energy, they can go farther. All right, so let's talk a little bit about endolymph and paralymph. So, paralymph, we know para means outside. Paralymph will be out here in the bony labyrinth. Endolymph, endo means inside. So endolymph will be in the membranous labyrinth. One of the best mnemonics you guys could use for this section is napkin. And I'll write it right here. And I wrote it like this for a reason. Na. Na is sodium. And it's next to P. You have the highest concentration of sodium in the paralymph. K is for potassium. You'll have the highest concentration of potassium in the endolymph. So napkin. All right. Going back to this tympanic membrane, out here is the pars flaccida. Inside, where the actual eardrum vibration happens, is the pars tensa. We've got a couple minutes left. All right. Are you guys good on this so far? Good. This was mainly the cochlear system. We're going to talk a little bit about the vestibular system. All right. So let's say I ask you about linear acceleration. What cells will you have detecting that sort of acceleration? So I heard someone say saculi. Yeah, so the type of cells are actually macula cells, and they're located in the saccule. So macula, saculi. For linear acceleration. You have more macula cells, but located in a different area in the utricle. So you have macula utriculi. These will help detect linear acceleration. All right, so we're going to throw in angular acceleration. For example, you turn really quick. These types of cells are called cristae. So what about the location? What's the rest of this name? Yeah, awesome. Cristae ampullaris. All right. It goes into a little bit of detail regarding the types of hair cells you have in the vestibular system. So we're going to introduce the topic really quick. It's not the most important, but it's pretty good to understand. So you saw structures that look like this in your textbook. This tall one is very important. This tall one is called the like kinetics, cilium, kinocilium. These other ones are stereocilium. There we go. Stereocilium. We have depolarization of the vestibular portion of the vestibulocochlear nerve when the stereocilium come in contact with the kinocilium. So when you accelerate in the otitis media, which is not that important, you guys don't need to know that. When the otitis media fluctuates and pushes the stereocilium up against the kinocilium. That's what causes depolarization. All right, we only have a couple minutes left, so I'm going to ask you guys 2 more innervations questions. We have 2 pretty important muscles in our inner ear. We have tensor tympani, and we also have stapedius. What's the innervation for tensor tympani? Cranial nerve 5. Can you get any more specific? Yeah, good. Cranial nerve 5, branch 3. The way I always remember this, T, and you add in trigeminal, T. So these T's will help you figure out trigeminal. You have 3 T's total. So branch 3 of trigeminal nerve. All right, so what about the stapedius muscle? Awesome, cranial nerve 7. S7, stapedius. All right, so we're coming to the end of our session. Do any of you guys have any other questions about this? All right, one other announcement I want to make. If you guys go on, and I'm actually going to switch to the desktop to show you this. All right, so if you guys go to a web browser, you can go to SARC's website. Here, SARC has a very important section for the study union. When you see this time schedule, go to this link. I highly recommend it. The link, I'll go back so you guys can see it. The link at the very bottom of the schedule, if you click it, you can actually go to this webpage where you can watch the study union session from today and from previous SI leaders. I talk about this in my SI sessions a lot, but for example, there are 6 other videos of this review, all emphasizing different topics. I posted a very large study guide, but I didn't touch it at all because about 3 videos ago, which is like 3 or 4 semesters ago, 2 SI leaders before me destroyed that review. That is all they did, and they went into insane detail. So you guys have this resource. I recommend you use it. Each video is 2 hours, so don't try to watch all of them at once. Space them out. And I also want to ask you guys to go on SARC's Facebook page and please review my session. You can just go on Facebook, UCF SARC, and it's going to be a link in one of their posts. I thank you guys so much for coming out and really have a good test. Good luck on it. I hope you guys do well.